I make it about 1:28, and so I think we'll do the introduction so we don't take time from the speakers. Welcome to this Grasslands Innovation sponsored session. I get to thank my small organisation for sponsoring it, so thank you. The session that you're going to hear today is called Plantain, a Journey from Weed to Delivering Multiple Values in Farm Systems. And I think this is quite a unique session at this conference in terms of a session that will cover from discovery and the original ideas that came to play of where Plantain may provide a role in farming systems to provide environmental benefits, right through all the research to where ultimately you're at the adoption end of this, and how do you make this work on farm and at a catchment scale. And so that's the range of papers that you will hear. And so it has involved a lot of work of various research organisations, and you'll hear presentations from 5 different organisations this afternoon. So without further ado, I will invite Ina Pinksterhouse to come up and give the first presentation. Ina had got her PhD from Wageningen University, and she has been doing research conducted after doing research at AgResearch Grasslands. Then she went back and worked at Wageningen Livestock Research before returning to New Zealand 12 years ago to work for Dairy NZ, where she has been working on participatory research and on co-innovation, of which this is a key part. And she led a large program called Forages for Reduced Nitrate Leaching, which was a precursor and a key part of leading to what she's going to now present. So thank you, Ina.
Thank you very much, Derek. Yes, my presentation is very much an introductory presentation of this series of 6 from the Plantain Potency and Practice program. You see at the bottom collaborators and funders. But very much my presentation is about the initial work done in the Forages for Reduced Nitrate Leaching and greener pastures. And again, you see all the collaborators there, so really quite large programs of work in New Zealand. First of all, a short introduction on what plantain we're actually talking about. We're not talking about bananas, we're talking about a broadleaf herb. This was commercialized over 20 years ago in New Zealand to use in forage pastures. And the types that are bred are more productive than the wild types. They're more upright, so grazers can harvest more of the leaf material, and they're more winter active as well. Compared with perennial ryegrass, what we see is they're producing more in summer dry conditions over the summer and early autumn, so they have been used in non-irrigated pastures for quite some time. They also have higher trace elements, micronutrients levels than perennial ryegrass, and also secondary compounds, and you will hear quite a bit more about those. Also, it has a higher water-soluble carbohydrate crude protein ratio, which has got benefits for nitrogen efficiency in animals, and a lower dry matter percentage. So water intake is greater with ingestion of plantain. However, it is also less persistent than perennial ryegrass and white clover. So this is something that farmers really need to get their head around. It is not a perennial species. It lasts 2 or 3 years, especially in the productive pastures. So we started to get really interested in this pasture species because of the properties I just mentioned. Because our farmers in New Zealand need more options to reduce nitrate leaching from their systems. As you can see on this map, large areas in New Zealand are over-allocated for nitrogen. So regulation has been developed with national targets, and regional councils at present are developing new regulation and rules for farms. And for some regions, that means that the nitrogen load from the farming systems may have to reduce by 80%, which is really quite massive. So we really need options that are practical. Now, in our grazed farming systems, grazed pastures, the main direct source of nitrate leaching is the urine patch. So a lot of our work has been focused on urine excretion. The normal ryegrass clover pastures have high crude protein content, which is in excess of what the animals require. So the excess is excreted in manure a little bit, but especially in the urine patch. And in our systems, the nitrogen load in those urine patches is commonly between 600-700 kilos of nitrogen per hectare equivalent, which is, of course, way above any requirements of the pasture underneath. So Plantain appears to be a really good option to look into because it's got several properties that will help us. First of all, there is a dilution effect. This was measured already over a number of years in several trials. The nitrogen concentration in the urine excreted by animals grazing plantain is quite a bit lower than that from ryegrass. At first we thought it was perhaps because of the nitrogen content, but that's not the case. It really is a dilution effect with greater urine volumes seen in the animals grazing plantain. What we also see is a lower proportion of the excreted nitrogen ends up in the urine. More goes into feces and a lot of the time also in the milk. There are other effects as well, and they are really located in the soil. It's about nitrification inhibition, so slower rates of nitrate production. These can be indirect through the urine or direct through the plant. And I will show some results from the initial work. What has also been measured is that the co-benefit of this is that also nitrous oxide emission from these urine patches is reduced. So first, an example of the dilution effect. You will hear more from Alina Menet later on, but this is a graph from her initial metabolism stall trial. So clear you can see with an increased proportion of plantain in the diet, the nitrogen concentration of the urine is lower. But also, clearly, there is a diurnal pattern here. And that was a really interesting and also important learning because a lot of the earlier work was with spot samples of urine. So you really need to know at which stage in this pattern you're actually sampling. Other work with sheep grazing showed that there's also a difference between this urine volume and nitrogen concentration between cultivars, which was an important result because it means that we cannot say that all plantains are equal. From the same metabolism stall trial, you see here an example of the partitioning effect. Coincidentally, in this trial with the different proportions of plantain, the nitrogen intake was very similar across all treatments. But you can clearly see that the proportion excreted into urine was lower with a greater proportion of plantain in the diet. That other nitrogen was distributed between feces and milk. And of course, as we see in most metabolism stall trials, there's a little bit unaccounted for. What you see on this graph is that correlation between the excretion of nitrogen to the urine compared with the carbohydrate protein ratio. So in this trial it was shown as well. This is something that is commonly known with TMR as well. So this graph is an example of the nitrification inhibition. The top line is from urine excreted by animals grazing ryegrass. And as you see, over time, the conversion from ammonia to nitrate is pretty quick. And the middle line is from the agritonic from one of the cultivars of Plantain, where you clearly see a delay in this conversion. By day 35, the amounts are pretty similar. So it is a delay in the nitrification, not a complete stop. It was also shown that these kind of delays are actually quite variable. It depends on how much plantain those animals have ingested and also over how much time. And again, differences between cultivars. If we combine these effects, we see a very big reduction in nitrate leaching, as you can see in this graph. These are nitrate leaching results from lysimeter studies. So lysimeters are undisturbed soil cores, large cores, where then urine was applied. Our control was without urine application, and you can hardly see the bars. There are little stripes at the bottom, so hardly any leaching if no urine was applied. The blue bar on the left is ryegrass white clover growing on those lysimeters, and urine from animals that were grazing ryegrass white clover was applied. That urine was equivalent to a nitrogen load of 664 kilograms of nitrogen per hectare. The striped bar is, again, ryegrass white clover urine, but then standardized to 700 kilos. So those two obviously are very close, and you see a high amount of leaching from those urine patches. But what happens if the sward was Italian ryegrass, white clover, and plantain? Plant soil effect here is with standardized urine applied, a 45% reduction in nitrate leaching. But if urine was taken from the animals that were grazing these Italian ryegrass, plantain, clover swards, the reduction was a whopping 89%. So it's very clear that there is a plant effect, but also an indirect urine effect. But still we have implementation challenges with plantain. Sometimes farmers report issues with palatability, that there are times of the year that animals just avoid plantain, which is of course not very beneficial to the effects that we want to see. And also, of course, weed management becomes more tricky if there is a herb in the pasture as well. So there are only a few herbicides that are available that can be used. Again, decrease of plantain content over time. Farmers see that as an issue. They have to continue to introduce plantain. So we did some work on that as well, on what methods can you use to do this. You get the highest proportion of plantain with a complete regrassing, but of course we don't want to do that every couple of years because that defeats the purpose of reducing nitrate leaching. So what you see here is some results from a plot study, and we did further studies on farms as well, comparing broadcasting and direct drilling of plantain seed, and also different management after the sowing. So in general, what we see is that direct drilling will result in a higher proportion of plantain compared to broadcasting, but also that an early grazing is actually beneficial. Usually you would think you need to let the plants establish. But of course with plantain in an existing pasture, there is competition from the grasses. So if we graze it early, before the plantain plants are above grazing height, we actually knock out some grass, competition is lower, and the establishment is better. But again, we see region-specific results here. and very variable results as well, so weather-related, grazing management-related, but also which pasture species are more prevalent and how competitive are they against plantain. So in the new Plantain Potency and Practice program, we are working on a couple of these aspects to understand better what is happening with plantain and also how we need to manage it. We're working on quantification of that nitrate leaching effect at scale under grazing. You will hear more about that as well. We try to understand better what those soil processes are and how plantain impacts nitrogen and carbon processes, so the mechanisms that reduce nitrate leaching. Because there are cultivar differences, we need to understand what they consist of, and are there actually traits that we can measure in plants or in the soil that will indicate if that particular plantain cultivar is effective or not. If we are asking farmers to use more of the plantain, we also need to know if there is any effect on our milk quality parameters, but also processability, for example. So we're investigating that as well. I mentioned before already management strategies to maintain plantain or reintroduce plantain in pastures, looking at grazing aspects as well. And all in all that work needs to come back to the farmer, of course. They need to become successful in implementing plantain so that they will have viable and practical solutions and are still able to farm under these new nitrogen limits. Thank you. Yeah.
So Ina has done so well, there is 5 minutes for questions. Over here.
Thank you. What's your experience with the maximum defoliation events per year? How often do you use the plantain per year?
That depends, of course, again, how productive the pastures are, but in general, the pastures are utilized the same as for ryegrass white clover. Yeah, so in our New Zealand systems, that's 11 cuts a year, or grazings a year, 10, 11.
Lindsay?
So, can you tell us about the environments where plantain is being used in New Zealand and where its limits are in terms of its adaptation?
It's utilized everywhere, across the whole country. The persistence under wet conditions seems to be lower. So that's Southland, for example. Those farmers are complaining quite a bit. So treading damage appears to be detrimental for its persistence. But plantain can cope with all the climates that we have across the country.
Another question?
Anjo, yeah.
Was the persistence in general, how many years would it last?
So
Well,
the question
under-
was about persistence, how many years does it last?
Yeah. In productive, highly productive pastures, 2 to 3 years, but sometimes even after sowing, farmers complain that there is not that much plantain coming up, and in other circumstances, they've got a good strike. So it's highly variable. What we're hearing from the dry stock farming is that it reseeds itself and it's persistent. Yeah.
What seasons have you found it's best to establish?
That's regional as well. Glenn, do you want to say something about it? I think we've used autumn and spring sowing. But
Stand up, Glenn, so everyone can hear you.
Yes, either. I think that would depend on the environment's going into. If you are sowing in the spring and it's relatively temperate, with that, if you're sowing in the spring and you get hot very quickly, it's probably also. If you go cold very quickly, then it's probably spring sow. So it does depend on go
And also in general, the real result of the resowing or the undersowing or oversowing is seen in the next season. so you don't immediately see it coming back.
Gavin?
What is the origin of this plant, and is it likely to move across into subtropical conditions?
Do you want to
Well-
direct that to Alan? Do you want Alan to answer that
I
one?
think he'll be the perfect person to.
Well, Plantain in its natural habitat is a Eurasian central, North African central origin, so it's a matter of getting in there. We think the material that we've got is probably down in the Spain-Portugal region, so a mild winter region with a little bit of rain in the summer, we don't have dormancy. And yes, in some of those regions there are subtropical plants, so we're finding that it can go into, obviously it's reasonably comfortable in Northland with like type climates, so, but yes, it is a roughly heat tolerant plant. And there's various forms that can be roughly winter hardy as well. It goes right up to Canada here in the United States, and often it's from the region. And it's certainly found in the southern states where it's a lot of heat. So it's a matter of breeding the right conditions.
So the question was, could it go into subtropical environments? So wasn't that what I heard, Kevin?
Trying to get an idea of what
Yeah.
our business is, a wide kind of
Yeah.
regions, competitions.
Well, certainly, that work in which is
Cancel. Start again.
It's not on here.
Can we grab one of the guys from across the aisle?
Let me see if I can do something.
Oh, it's coming? Oh, it's good. All right.
I don't know what I did.
Thank you. Self-fixing. Need the experts. Good. Our next paper is by Gemma Box, who's a scientist at DLF. She's specialising and actually leading the forage herb breeding, including plantain. She has a special interest in improving the environmental traits of commercial species, and her PhD was actually in large animal nutrition, and which broadens the elements and influence of that on the breeding program. So all yours, Gemma.
Thanks very much for that, Derek. So today, as a plant breeder, I'm going to talk about the development of environmentally functional plantains. So just during the session, I'll give just a really brief overview, and I gave a great introduction of plantain use in Australasia, the commercialisation of plantain, and then finally how we developed a cultivar and our future breeding cultivars have environmental functionality. So plantain use in Australasia, it's a herb species, and it's typically grown as part of a diverse pastoral sward at around that 10 to 30%. There is some use as a monoculture, especially in lamb finishing systems. Cultivars such as Ceres tonic, we typically expect an annual yield of 10 to 15 tonnes of dry matter per hectare per year. Its quality sits around the 11 to 12, quite variable dry matters depending on the season, and a crude protein of 16 to 28%. So plantain was one of the few species that was commercialised last century. It's quite a ubiquitous flatweed naturally, and it was bred, Alan Stewart actually bred an erect winter active cultivar called Ceres tonic around 25 years ago. The sales of plantain seed throughout New Zealand is around 240 tonnes of seed sold per year. So what are environmentally functional plantains? So these are ecotains with known functionality. Agritonic was the first ecotane type but we do have several breeding lines in the research and development program. So that's our base standard, and everything else we make sure that has that environmental functionality. When we were breeding for agritonic, we had 4 main objectives. We wanted to maintain the yield performance and seasonal distribution, which means it's winter active, of ceres tonic. We wanted to increase the competitiveness within the sward. We wanted to provide herbicide options for weed control, which is especially important in a diverse pastoral sward with ryegrass and white clover. And we wanted to elevate environmental functionality. So the first target, maintaining the yield and performance. When we do our visual scores, there's no visual differences in growth between tonic and agritonic throughout the year. And in our plot trials, there's actually no significant difference in yield between tonic and agritonic at around that 10 to 11.5 tonnes of dry matter per hectare per year, respectively. Our second target was to increase the competitiveness within a sward. So we did that by breeding tonic with a narrow leaf, highly tilled type. So agritonic has the same leaf length as tonic, but it has increased leaf density through its tiller numbers as well. And agritonic is also much more erect than tonic too. So we get a really dense and upright plant, which is likely to increase the competitiveness within the diverse pastoral sward, especially with some of our premium grass products that really grow quite quickly. The third challenge that we really wanted to overcome was to provide farmers with some herbicide options. You can imagine with multiple species in a sward, it is quite challenging to get those herbicide options. So we did this over 5 cycles of selection using MPCA and 2,4-D amine. So you can see the 2 red circles. The ones that are on the left-hand side, they would be selected to move forward in the program, whereas the ones on the right-hand side would not be selected. So this is actually an off-label claim, so all the plantain is affected, but what we were really selecting for is really fast regrowth after these herbicide spraying events. If we compare some of the breeding program, line 1 has had 2 cycles of selection. Line 2 has had 8 cycles of selection under herbicide under our herbicide regime. Line 3 has had 2 cycles, and line 4 has had 7 cycles. So you can really see the difference that we can make over a successive cycles. Just for any non-plant breeder, a cycle is where we spray it, we then select those plants that we want to, we cross those together and then do another generation. So that would be 8 generations in line 2. The most important was we really wanted to elevate the environmental functionality. We really wanted to add value to farmers with this product. So our elite material that has already achieved the previous 3 targets enters into the final stage of selection, which is for favourable plant chemistry traits. So we're either selecting for the presence or the absence of a number of chemicals or secondary plant compounds which have been identified in the literature. And we do this through performing measurements on both leaf and root exudates. So we always recombine the best plants within the best families for successive generations, and we can currently recombine the worst ones as well to ensure that we're making progress. So if we take this example here, this is of one of our chemical traits after 4 rounds of selection, and we take if you imagine a bell-shaped curve, we take the 2 extremes and recombine them. So you can see those 2 poles really being pulled apart there. So our ecotains have 4 modes of action. and the rest of this presentation I'll talk about those modes of action. These modes of action significantly reduce nitrogen leaching from the urine patch and also reduce nitrous oxide emission, and there's also emerging evidence of changing methane production through a difference in volatile fatty acids. So our four main functions, we have delay, reduce, restrict, and dilute. So if we think about our nitrogen cycle, hopefully we've all seen this one before. So what we're talking about is the conversion of ammonia through to nitrate, which then gets leached through the soil profile, especially in water events. So our two animal functions, the reduce and dilute, they act on the animal, and our two functions, delay and restrict, act in the soil profile. So dilute, this is where there's a diuretic action directly on the animals. So the animal eats a large amount of pasture, takes all that nitrogen that it's collected and excretes it in urine patches. With a diuretic, you imagine the same amount of nitrogen being captured, but if there's a diuretic present, each urine patch, it weighs a lot more, so each urine patch contains less nitrogen overall. So this here, both these lines here are plantain cultivars. The one on the right, the far right-hand side, that is one of our ecotains, and the one on the left-hand side is not. So you can see a difference within cultivars in terms of the diuretic ability. The next function is the reduce function, where agritonic alters nitrogen partitioning, with a higher amount going to fecal material compared to ryegrass diets. So in the rumen, spare ammonia quickly gets absorbed into the bloodstream and then gets excreted as a waste product in the urine. And we think that this is affected because of the low solubility of protein with plantain, high carbohydrate providing microbials with a lot more energy, and a lower rumen retention time. So a couple of papers, one showed a 40% reduction in rumen ammonia over 24 hours compared to chicory, and another one showed a 33% reduction in nitrogen than those grazing ryegrass and white clover. The next function is the reduce function. Oh, sorry, that was the same one. The next function is the delay function. So what this does is it delays the conversion of ammonia to nitrate in the soil profile in the hope that other companion species will take up that nitrogen and use that as plant growth rather than getting excreted through the soil profile. So agritonic urine patches are slow release, therefore allowing greater plant uptake. So as Ina pointed out, one lysimeter study at Lincoln University showed an 89% reduction in nitrate leaching when animals consumed plantain, and then that was excreted into a plantain-based sward. So we do selection of our elite breeding material, which consistently reduces nitrate leaching. We recombine that. And the final mode of functionality is the restrict, where we think because of the presence of a biological nitrification inhibitor in the roots of plantain, it restricts the conversion of ammonia to nitrate. And this differs between plantain cultivars as well. In terms of what we do with the restrict function, we do measure the root exudates of single plants in our elite populations, and then we recombine single plants with the highest concentration of root exudates. So it's pretty clear that plantain can be really useful in the nitrate leaching side of things, but the combination of all four of those modes of action can also act to reduce rumen and therefore urinary nitrogen as well. And there is emerging evidence of lower methane production. So in summary, our creation of agrotonic and future elite ecotains involves maintaining or improving yield, increased competitiveness within the sward, increased herbicide options for farmers, and elevates environmental functionality. And where we see this as a good fit is it really adds value to the farmer, particularly with a reduction in nitrate leaching and nitrous oxide emissions that in New Zealand can now be accounted for in the program overseer. The addition of ecotain also expands the biodiversity within our pastoral swards. And feeding ecotain increases the feed diversity, which supplies a different number of metabolites available to ruminants, which can act as anthelmintics and have antimicrobial properties too. So that was the development of environmentally functional plantains. Are there any questions?
As with Ina, you've given us plenty of time for questions. So where do we want to start? Over here. What kind of cultivars, commercial cultivars, can we expect in the next few years?
Sure, so this is our baseline, but we do have a late flowering cultivar that's coming to the market. So if you can imagine, plantain starts flowering in springtime and continues all the way through to autumn, I guess over a 6-month period. So what we've done is we've taken when that starts flowering to a little bit later, so that therefore it uses less of its energy in reproductive growth and more energy into vegetative growth.
You can write that down, Pete. Lindsay.
We talked about persistence as a challenge. Did you add that to the selection criteria going forward, and how do you leave that vote?
We definitely do have part of the program focused on persistence, but it's always difficult with the addition of any traits, especially persistence traits. If we want to have it persist for, say, 10 years, but we might want to run 5 selection cycles on that, that's 50 years that you have to have that in the ground for. So that it's definitely something, part of the program that we are working on, but it's a long game fix, yes.
So Gemma, just follow up on that. Even if we were to select for greater persistence, are we going to end up with a true perennial, or are we going to end up with an extended biennial? What do you think?
Yeah, I mean, it naturally fits in that 2 to 4 years. So maybe we should just focus on traits that are good in that time frame, especially the environmental traits.
Over the back.
In terms of breeding plantain in order to reduce nitrate leaching, is there a trade-off to be made in terms of management? You did mention there that a more extensive system, plantain does persist quite well because it can self-seed. So would it be, in the immediate future, would it be more advantageous to change the dairy system in New Zealand, reduce stocking rates, be more extensive and include the plantain, or reduce nitrate leaching overall?
Do you want to paraphrase that back for the audience, Gemma?
Sure, I'll let you do that, Terry.
So you're basically asking, is there a trade-off between the breeding for the functionality versus how it fits, versus, say, just reducing stocking rate instead of breeding for the trait?
Yeah, would that help with your plantain resistance?
So we always fear undergrazing rather than overgrazing in terms of plantain. When we run trials side by side, one's under lax grazing and one's under really strict grazing regime. The ones that are under lax grazing, the older leaves become less palatable for the animal and they accumulate disease, which then goes down into the root, and we actually find that to significantly reduce the persistence rather than a fast grazing rotation. sorry, what was
How do you account for the better persistency in the beef system? How does it persist better in beef pastures rather than in dairy systems?
Oh, sure. So I maybe Ina might be better to answer this, but I think it's to do with the pugging and the more intensity on the soil type.
And more natural reseeding opportunity in those beef pastures. That's what you said. Yeah. So in the beef systems, it's the reseeding that's providing the longevity. The plant isn't necessarily going from being a 2-year plant to being a 4-year plant. So you're getting the next generation of seedlings. Soledad, why don't you come up and, well, we've got time for one more question, but if we change the presentation while you're answering the question, we've got time for one more. Oh, come on, I haven't read the question. You had them on the tip of your tongue before, so one here.
I was wondering, is the concentration of these secondary metabolites correlated to some phenotype or trait you can observe? Because it's challenging for us to do it in the lab all the time. And I read that it accumulates near the leaf nerves or something.
I'm not sure that you're allowed to answer that one. If she answers that, we might have to kill the whole audience. So. Is that all right? Thank you. Let's thank Gemma again. So we've got about 20 seconds till the next speaker. So I will do my best to not botch her introduction. So, Dr. Soledad Navarrete, who's originally from Chile, is a research officer at Massey University with knowledge of animal and pasture sciences. In 2015, Soledad completed her PhD at Massey University, looking at the agronomic performance of plantain pastures for New Zealand dairy systems. Since 2016, she's continued her work on evaluation of plantain as a mitigation option for New Zealand pastoral dairy systems to reduce their nitrogen losses. Sole, you have the floor.
Thank you, Derek, for the introduction. Good afternoon to everyone. Today I'm here with this presentation with evidence of the environmental benefit of plantain pasture. The previous presentation were very clear and provide a good understanding of the mechanism by which plantain can reduce nitrogen leaching from urine patches. But in pastoral system, where the urine from the cows is the main pollutant or the main contributors to on nitrogen leaching, quantify the effect of plantain reducing nitrogen leaching via direct measurement or at paddock scale level, and understanding how plantain works to reduce the nitrogen losses at paddock level and the key factors influencing these losses is the evidence that science needs to provide to give confidence that plantain really works and will provide a solution or a mitigation option for pastoral dairy system or all pastoral system, if we can later say that. And in the past years, at a dairy system at Massey University has been doing this part of the work. We have been focused on evaluate the productivity and the nitrate leaching from plantain-based pastures in a large dairy system experiment for 2 years, but it is an ongoing research, so we are now on our 4-year of evaluation. But I will focus this presentation basically mainly for the 2 full seasons that we have complete. From 4 pasture treatment of perennial ryegrass white clover, the traditional sward, and incorporating here different proportion of plantain, targeting 0, 30, 50, and 70% of plantain in the dry matter or of content in the sward. The research site where we have been evaluating this pasture consists on 20 experimental plots with their own drainage system. This plot has a mole and pipe drain system and are individually hydrologically isolated. And this picture, it's our experimental site. It's a 7-hectare paddock where we have 20 plots that are grazed with dairy cows for the full production season. And we have also incorporated 4 additional areas of around 1 hectare each one, where we establish one of each pasture treatment, so we can have a period of adaptation before or prior we allocate the cows to graze on this drainage plot or drainage experimental plot. We all collect, we have the sampling stations at the edge. It's on a slope here, so here where all the water from each plot drains, and we can collect and subsample for later analysis of the nitrogen, the nitrate concentration and the total nitrogen as well. We established the 4 pasture treatment using 5 plots. So we have 5 replicate experimental plots for each one of our treatment, and the 4 additional, and one of the 4 additional areas also for each pasture. So we have the 0% plantain or the traditional or our control, the ryegrass white clover. The outside area is located on this part. Then we continue the next area around the experimental plot is the 30% of plantain, and we continue with our 50 and then our 70% of adaptation area. How we manage this treatment over the years, it's basically grazing from spring to late autumn winter. Around, in this trial, was around 8 times each year or each season. We have been over 2 years, and I will say like spring start from spring 2019 to autumn winter 2020, and then we continue for the second year 2020 and to the season until winter 2021 has our year 2 in terms of grazing management. We use always the same group of cows, and the number of cows we use vary according to the availability that we have of the herbage mass in the pasture, but always was between 15 to 20 cows in each pasture type, and which allow us to allocate 3 to 4 cows per plot to graze during at least a minimum of 2 days. And the time that cows spend in this plot was between 2 to 3.5, also depending on the availability of the pasture mass in each treatment. But we did all the management under the same conditions, so we came with the cows first adaptation 6 days outside, and then they came on day 7 into the experimental plot for until day 8 or day 9, roughly to say. And then you can see here in this picture how cows are grazing the experimental plot. And we remove also the animals at the same time. So everything, the management, we didn't did much changes. We keep all the conditions very controlled in that terms. And then to be able later in winter, especially, to collect and monitor all the nitrogen that was coming out from each of these experimental plot over 2 years. So we have our winter, spring, the rainy season from 2020 as our year one, and then on winter 2021, we collect a second season of drainage water, which we analyze the nitrate content, concentration. We took quite a lot of measurement, and the key result from this trial, and what most people want to know, is nitrogen leaching at paddock scale. And how consists our direct measurement? Basically, here we have our sampling point for each experimental plot. So one of these one is where comes the water from one plot. So this is one plot, a second plot, a third plot, or fourth plot. We monitor the volume that runs through the drainage season, and we have here our sampling collection from a representative proportion of water coming from the plot. So we estimate the concentration when we sample. We sample different drainage events, usually around 20 millimeters of rain. It's roughly when we need to go to the sampling point to check and collect our sampling. We took only 100 milliliters of drainage water, and that one is the one that we analyzed. And then we just transform that to the total amount that it's accumulated through the season to know the cumulative nitrate leaching. At every grazing, we always monitor and take samples for pasture yield, so we can know at the end of the year how much productive were the pasture with different proportion of plantain. But also we want to know the proportion of plantain and how this changed through the year, and how much, of course, is across the whole season. We also monitor milk production and evaluate the composition, and we take more detailed measurements during summer through autumn. That's the period where we focus our details analysis on the milk composition, are in also from the urine nitrogen concentration, the urine N from spot samples. This is a grazing experiment, so that's what allows us to do the best way that we can know the nitrogen that is coming out from the urine. But we also took some additional measurements so that we could understand a bit more how plantain is working at paddock level, at paddock scale level. And one of the experiments we did, we evaluate the urination patterns. So that's basically urine volume and frequency every time that the cows was peeing, using urination sensor that we attach to the cow's vulva. And we did this one on 2 grazing rotations on autumn. And we also take the opportunity to measure nitrous oxide emission from the urine coming, urine patches in this case, during late summer, autumn of the second year of the experiment. The 4 pasture treatment, where we targeting the our control with 0, 30, 50, and 70% of plantain, were established using different sowing rates. We reduced our rhizus seeds rate from 20, 15, 10, and 5, as you can see on this table. We kept white clover with the same rate. We used 3 kilograms per hectare. And the plantain sowing rate used in this treatment was an increasing level. So we increased from 0, and we used 4, 7, and 10 kilograms of plantain seed per hectare for our 3 plantain ryegrass based pasture. In the first year and during the establishment year, we observed that the plantain in our 3 pasture was 30, 42, and 50%. Later on the second year, and after adding more plantain seeds into each of the plantain treatment, we used 3, 6, and 9, basically to keep our plantain content. We know from previous experience that plantain, and also from the first year, that plantain increased during the establishment year. And from previous research experience, we know that the decline of the plantain population or plant density or composition in the pasture is after the second year. So we try to keep to our best the plantain content in the system, so we can get a range of different proportions. But in the second year, the plantain proportions this is the average across the whole year or the whole production season, so from spring till winter or when we complete the grazings for each year, and was 30, 47, and 45%. We learned from these first 2 years that sowing plantain with rates between 4 and 7 kilograms per hectare allow us to obtain a proportion of plantain between 30 and 50%. However, we were unable to obtain a high proportion of plantain or a plantain treatment with at least a 70%. We basically have 50% was our threshold in between combining ryegrass and white clover. Sorry, okay. And the cows produce similar, the pasture production was similar and the milk production from the cows was also similar. But indeed, what plantain did is reduce nitrate leaching from grazed pasture by between 20 to 60%. This pasture, I mentioned in the previous slides that in the first year we are talking about 30 to 50% of plantain, and then 30 to 45% of plantain. We have difference between year 1 and year 2 because environmental conditions change this, and also on the first year, the establishment also of this drainage plot is more disruptive, and also it takes soils build up nitrogen over time. But it's clear that there is a significant effect of plantain reducing nitrogen leaching. One of the changes that it was also mentioned before is that cows grazing of plantain produce a urine with a lower nitrogen concentration, reducing in this way the leaching from nitrogen leaching from the urine patches. Was clear that you can see here all the bars on blue represent our plantain ryegrass treatment, and we have a clear effect of plantain reducing by 30 up to near 40%, depending of the plantain content and the season. We wanted to understand how this is working on the cows, how cows are changing the urination patterns, or what changed on the animal when it's grazing. And we focus our measurement on autumn on the second year. And we learned from this study, that's a small study or more focused research, that plantain in the cow's diet indeed increased urine volume, as mentioned in the previous presentation, reducing the nitrogen concentration via a dilution effect. But what we can see also that increased the urination frequency of the animals. So cows are peeing more frequently than cows grazing on on ryegrass white clover pasture, which means that the urine nitrogen is spread across the paddock at lower nitrogen rates, but per each urination event.
2 minutes.
Yep. Plantain has the ability to reduce the nitrogen loading, and this is basically how it's working. Over 2 years, if we focus on this figure, I want to highlight from here that when we evaluate how change the urine concentration against the urine volume, this was the mechanism effect was working at all time. But when we focus on see how was the total nitrogen coming out from the urine of the cows, the research is telling us that this is happening from February, March, on May, which in New Zealand or in the southern hemisphere is from late summer to autumn. Our key drivers reducing nitrogen concentration in the urine is because plantain has a lower dry matter content than ryegrass. So cows are drinking more water while they are eating the pasture. There's also higher mineral concentration. We think sodium is also a driver that increase volume. And we are also thinking on the secondary compounds that plantain contains can have a diuretic property, so increase the urine also volume. And our key drivers decreasing the nitrogen content or the total excretion via the change in the nitrogen partitioning. It's the higher non-structural carbohydrate in plantain, which also translate in a higher non-structural carbohydrate to nitrogen ratio. But also we have secondary compounds that are doing their job here and shifting or changing the partitioning. And I will skip, but also decrease nitrous oxide emissions. And basically I'm gonna conclude that incorporating plantain in ryegrass white clover pasture reduced nitrogen leaching by up to 60% in this study at paddock scale level and reduce nitrous oxide emissions. The greater spread of the urine at lower nitrogen loading rates per urination event is what is telling you how it's plantain working at paddock level or in grazing pasture. And this combined with the soil swap effect that we will hear later is also influencing the nitrogen conversion pathways and indicates the potential of plantain to reduce nitrogen losses at farm scale level.
Thank you, Sally.
Yeah.
Right. I know there'll be a lot of questions out of that in terms of we missed a little bit of the nitrous oxide reductions as well, but in keeping on time, we're going to move to Michelle's presentation. So if you can change those, that would be great. So, Michelle Peterson is a scientist at Plantain Food Research, working in their crop and environment unit. Her PhD was as far from agriculture as you could get, characterising enzymes isolated from microbes that live in volcanoes and hot pools. She is an analytical biochemist who now works in soil and plants, and she is active across several research areas, including the one she's going to talk about today with biological nitrification inhibition in cereals and in forage crops. Thank you, Michelle.
Thank you, Derek. I want to start by acknowledging the funders of two projects from which I'm presenting work today, the Greener Pastures Project and the Plantain Potency and Practice Program. And I also want to acknowledge the huge amount of support in these projects from my colleagues over the years. So one way to reduce nitrogen losses from intensively grazed forage systems is to inhibit nitrification. So nitrification is the stepwise conversion of ammonium to nitrate. It's carried out by a specific group of soil microbes called the ammonia oxidizing bacteria and archaea. So inhibiting nitrification keeps nitrogen in a non-leachable ammonium form. This extends the window for plant nitrogen uptake. It allows soil microbes more time to accumulate their more favoured nitrogen form, and reduces the amount of potentially leachable nitrate and or emitted nitrous oxide. So synthetic inhibitors of nitrification have been evaluated in forage systems. They're expensive, they might cause plant damage, and there are concerns about carryover into food products, which is what we found in New Zealand with the use of DCD. However, there are plant species that contain chemicals that also perform this nitrification inhibition. We call this activity biological nitrification inhibition, BNI from now on. It's been identified across a range of plant species. So plantain is one of these species, and there's research to suggest that it may inhibit nitrification in several ways. By release of inhibitory compounds when you mulch the leaves back into the soil, by release of inhibitory compounds from the roots via exudation, but also the excretion of inhibitory compounds in the urine of animals that have consumed plantain. The compounds responsible for these displays of inhibition haven't been yet confirmed, nor has their mode of action. But all of this evidence put together suggests that incorporating plantain into the sward might be a potential tool for mitigating N losses from forage systems. So today I am going to present results from 3 experiments that examine urine and root exudate BNI. We hypothesised that the compounds in plantain specifically target those soil microbes that carry out nitrification, and that the degree of nitrification in both urine BNI and root exudate BNI is cultivar dependent. So in our first experiment, we looked at the effect of urinary excreted compounds on soil nitrification by way of an incubation that just uses soil microcosms. So sheep were fed either ryegrass or plantain for 14 days. We've got evidence to suggest that a minimum of 10 days is required to see this effect. So after 14 days, we collected spot samples from each sheep. They were combined. Because the ryegrass urine has a higher total N concentration than the plantain urine, was diluted so that we were adding the urines at the same N concentration to those soil microcosms. We also included a urea treatment at the same N concentration as the urines, and we had a no N control that just received some water. We extracted soil mineral N and DNA during the incubation and sequenced bacterial, archaeal and fungal amplicons so that we could compare the change in microbial community structure across time and between our treatments. So up to day 9, the conversion of ammonium to nitrate in the ryegrass urine microcosms tracks the same as the no N, so our control microcosms, but nitrate production in the plantain microcosms, which is not working. The black line here is our plantain, is delayed. So by day 12, this delay has been overcome, but the nitrate in the plantain microcosms is nowhere near catching up to the ryegrass or the urea microcosms. However, by 21 days, the ryegrass and urea are pretty much the same, but there's still a heck of a lot less nitrate in those microcosms that have been treated with plantain urine. If we look at what the addition of the urine to the soils has done to the microbial communities, we see 90% similarity across the 4 treatments for the archaea and 70% for the fungi. So time didn't have a big effect. But when we look at the bacterial communities, sorry, I'm just going to change something here. When we look at the bacterial communities, we see that the urines group together and the urea and the soils group together, but they're dissimilar to each other. although the community structure does migrate in the same direction with time in all 4 treatments. So it seems that dietary composition doesn't have a big effect on the structure of the bacterial community, but there's a clear effect there on the rate of nitrification in those microcosms. So this leads us to believe that there are indeed compounds excreted in the urine of these sheep fed plantain that target those microbes that perform soil nitrification specifically without affecting the soil microbial community as a whole. Moving on to our second experiment, we looked at whether this urinary BNI we're seeing depends on what cultivar has been fed using an animal trial and a soil microcosm incubation similar to what I've just described for experiment 1. Here we fed different cultivars after which urine was collected, added to the soil microcosms and soil mineral determined. We also isolated the organic fraction of each urine using solid phase extraction. So this is to get rid of the urea and salt components in the urines to get to where we think the compounds that cause the inhibition are in that organic fraction. And inhibition in this fraction was assessed using a bioassay specific for nitrification because we use a pure culture of a bacterial a soil ammonium oxidizing bacterium. We also determined leaf concentrations in each cultivar of 3 bioactive compounds that are thought to be associated with BNI. We know from previous research that the concentration of these compounds is cultivar dependent. So if they're the origin of the metabolites responsible for the BNI that we're seeing, well then differences in their concentrations in the leaves might be reflected in urinary BNI. So we found some significant differences in the amount of nitrate produced in the soils treated with the urine from the different cultivars. Like we saw in experiment 1, in the first 7 to 10 days, we saw delayed nitrification compared to our no urine added control. And again, like we saw in experiment 1, that delay is eventually overcome. This was around about 10 days for cultivars B, F and H, and then 12 days for the rest, excluding the one that was treated with urine from sheep that were fed cultivar I. And yes, it seems that the inhibition by the urine is associated with that organic fraction. The bioassay that we used showed a similar cultivar-dependent trend as the soil nitrification assay, with cultivar B inhibiting the least and cultivar I inhibiting the most. When we looked at the concentrations of the 3 bioactive compounds thought to be associated with BNI, we saw the expected cultivar differences in orcubin and verbascicide. We didn't detect catalpol. This may be due to seasonal variation. We simply sampled at a time when catalpol was not being expressed in the plant. When we correlated the concentration of these bioactives with nitrate produced after 28 days in those soil microcosms, or percentage inhibition in the bioassay, we found that there was actually no relationship with orcubin. We did see a loose correlation between verbascicide and both measures of urinary BNI for the few samples that we have data on. And that kind of gives us some clues where to start looking for those compounds that are actually responsible for the inhibition that we're seeing. In our third experiment, we moved on to looking at BNI in root exudates, and we looked to see if this was also cultivar dependent. We assessed exudate BNI in plantain cultivars growing in a hydroponic system. The cultivars were grown in discrete containers with around about 20 plants per container, and after 4 to 5 weeks, the plants were removed from the system and the root exudates collected and processed. We assessed exudate BNI using the same bioassay specific for nitrification I talked about before, using that pure culture of soil ammonia oxidizing bacterium. And we submitted the leaves, the roots, and the exudates for metabolomic profiling. We looked at the concentration, we looked for orcubin, catalpol, and verbascicide across those 3 samples. And we also used an untargeted approach, which is where we use mass spectrometry to comprehensively analyse for all measurable metabolites in each sample. So exudate BNI here is expressed as the percentage decrease in nitrification rate caused by the exudates relative to an uninhibited control. We saw variation between the cultivars. We also saw a pretty big variation between replicates of each cultivar, but despite this, it's clear that exudates in cultivar V inhibited less than those from the other cultivars, particularly agrotonic and cultivar X. So our metabolomic profiling showed that aucupin and verbascicide, they correlated with, in the roots, correlated with the exudate bioassay results, but aucupin and verbascicides in the leaves didn't, which is what we found in experiment 2. We didn't find either of these compounds in the exudates. We actually found 2 different compounds in both leaf and root material that better relate to BNI activity than aucupin and verbascicide. And we found several compounds in the exudates that seem to relate to BNI, one of which I show here. We don't have a clear ID on what these compounds are, although we do have an idea of the chemical class. And given the variation in the bioassay results, we need more data to prove if this relationship really does stand. So to summarise everything I've presented here, plantain root exudates and urine from sheep-fed plantain inhibit nitrification, and the degree of inhibition is cultivar dependent. The bioactive compounds thought to be associated with BNI do not themselves appear to be directly inhibitory, but it may be that compounds responsible for both of those modes of inhibition are metabolites of these bioactives. Their identity and their mode of action we still need to figure out. The plantain-derived nitrification inhibitors in urine are specific, don't appear to have an effect on the soil microbial community as a whole. And it's important to remember that what I've presented here are laboratory findings. It's going to be important to go to field scale trials to validate, including plantain and forage systems as a tool to mitigate nitrogen losses. Thank you.
Thank you very much, Michelle. That's excellent. Do we have any questions for Michelle? Right down the back.
Do you know how long is the inhibitory effect of these compounds in the soil, or how long they last?
Well, I could go back to that graph if you like. It seems to be cultivar dependent, firstly. But it seems that it's generally overcome. You've got to remember these are soil microcosms, no plants in the system, and we're seeing that overcoming of the delay at around about 14 days. Yeah.
Gavin down the back and then down here.
I'm aware that the same kind of biological inhibition occurs in some tropical grasses. Is it similar mechanism? What do we learn from that relative to plantain? Is it similar action?
Well, I guess the thing is, all of the research in this space has used that pure culture of an ammonium oxidizing bacterium to look at the amount of inhibition in exudates. The compounds are different. In sorghum, we have sorgoleone. Brachiaria, we have brachyleone, brachia something, I can't quite remember. The compounds that we're seeing in plantain, different. In other cereals, different. But they're all acting on the same process, which is quite defined. Sorry, I didn't quite hear that.
He said, is that specific to exudates?
Yes, that's exudates. Yeah.
Right. Down the front here.
So, since you didn't see changes in your microbial community, do you think it's possible that the compounds are actually acting on enzymes in the soil?
Yeah, absolutely. That's That's exactly what I think. We think it's a direct action on ammonium monoxygenase, but we can't get pure ammonium monoxygenase. I'd love to be able to purify it. It's impossible. But yeah.
Lindsay.
Maybe not a question for you, but what's the delay in the process actually mean in terms of the risk of reduction of nitrogen leaching?
Maybe that refers back to Sole better than this one. So, Sole, do you want to answer that? What is it? What does this process mean in terms of actual effect on reduced nitrogen and, in your case, also nitrous oxide in the farm system? Then we'll move to the next speaker.
So, who made the question? Okay. We have operating to. So. Yeah.
What's the benefit for reducing nitrogen leaching?
Okay. Yeah. Okay. That's yeah. Okay. So.
Sorry, what was the benefit of the nitrogen inhibition?
It's more at scale.
Okay. At paddock scale. Yeah. The beneficial effect from this one is telling us that that's the power of plantain. We have two effects operating, one at the animal, but on the soil, the influence of reducing the nitrification, it's reducing the nitrous oxide emission to the air, but also changing and reducing the emission factor. So the amount of the nitrogen emitted from the urine nitrogen is much lower. So the losses are totally decreasing, or being, how Ina mentioned, the retention, or Gemma talk is the delay and retention in the soil and keeping the nitrogen on the soil. Say something? Convinced?
Ina would like to add to that answer, so I will.
Yes, because it's a very good and important question, and I think that's for us scientists the big challenge. Can we actually untangle which of all these effects is the most important one? So we tried it with modeling as well. The EPSIM model is being developed for plantain, and we're starting to introduce these effects that we're seeing to see if the model will tell us a little bit about that. And indeed, with the lysimeter studies, etc., we have untangled the urine effect and the plant effect, and they both are important. But then from the urine, the dilution effect and the compounds in the urine, that's another question again. So there's indeed a whole series of experiments needed to try and untangle that. Thank you.
And in the end, isn't it, the cumulative effect is what we want. We like to understand the mechanisms, but how we get to the overall reduction, maybe it's the total of all those processes that's important. All right. Sorry, we'll now move to our fifth speaker in this session, Elena Mine, who is a senior scientist at Dairy NZ in Waikato, with a background in agronomy and whose current research areas are environmentally sustainable dairying, climate adaptation and greenhouse gas mitigation. Elena gained her PhD from Lincoln University, studying nitrogen metabolism of dairy cows fed different forage diets. So we'll hand over to you, Elena.
Thank you, Derek. Kia ora koutou. So today I'm going to be just digging in a little bit more on what you've already heard, but into the particular traits of the plantain forage that drive the animal response in reducing the nitrogen loading potential from cattle grazing pastures. Ina previously gave a really good background of the situation in New Zealand. You know, New Zealand has a very temperate climate that allows for the growth of pastures for the majority of the year in the main dairying regions. And the challenge that we have is that our permanent pastures are very high yielding, but they also contain a high level of dietary nitrogen, well in excess of what the animal actually needs. And as we all know, an animal cannot store that excess dietary nitrogen, and it is excreted largely in the urine. And again, as Ina said, this excreted urine often comes in very, very high nitrogen loading rates, surpassing the capacity that the plants can uptake that nitrogen, and that remaining nitrogen is vulnerable to loss to the environment, either as nitrous oxide, a potent greenhouse gas, or lost via leaching into the groundwater. So a common research focus has been looking at ways that we can reduce the nitrogen loading of urine patches, and through the programs of work that Ina touched on before, FRNL, Greener Pastures, and now the Plantain Potency Program, there's been a substantial body of evidence that's been gathered that relative to ryegrass, feeding livestock plantain reduces the nitrogen loading of urine patches. And there's three sort of main mechanisms through which the plantain can achieve this. The first is the ability to sometimes reduce nitrogen intake, reducing the amount of the nitrogen the animal actually eats. The second is through that dilution of urine that we've heard a bit about already today. And the third is by altering the partitioning of dietary nitrogen, or altering the efficiency of the nitrogen use of the animal. So I'll touch on these very, very briefly, and the first again was reducing nitrogen intake. It's very well understood that as dietary nitrogen intake increases, the amount of nitrogen excreted in urine increases. So dietary mitigation strategies have looked for forages that are lower in nitrogen content. When we conducted a meta-analysis of 34 studies looking at the nutritive value of plantain compared with perennial ryegrass, we found that generally plantain has very similar total nitrogen content to plantain, so this is not one of the main mechanisms that we have for reducing nitrogen loading. But importantly, we did see in autumn or fall in the States that we can see a reduction in the nitrogen content of plantain pastures relative to ryegrass in autumn, and this is a particularly important month to observe this effect, as in the autumn any excreted nitrogen is more at risk of loss to the environment due to reduced plant growth and increased drainage through the soil profile. The second mechanism that has been discussed today is dilution of nitrogen of urine. And the graph that I have presented up here is essentially accumulation of the studies, the livestock studies that have looked at feeding animals various proportions of plantain in the diet, and we see a reasonably moderate negative response of reducing nitrogen concentration as the amount of plantain in the diet increases. And another urine picture, which we seem to like, it's very, very obvious when animals are consuming a ryegrass-only diet, their urine is much, much darker than animals that are consuming plantain. This is from cows that were consuming 40% of their diet as plantain. And when we measure the solutes of that urine, it's also much, much lower. And one of the main mechanisms that we have for diluting the urine is increasing urine volume. So again, we look at a grouping of all the studies that we've done to date. As the percentage of plantain increases in the diet, we see a corresponding increase in the urine volume that animal is producing. So we actually conducted a metabolism stool study to quantify the effects that plantain was having on the nitrogen metabolism and urination behaviour. And what we actually observed was that there was a bit of a threshold response with the urinary volume, that when we fed above 30% plantain in the diet, we saw a substantial increase in the amount of urine that the animal was producing, so potentially a bit of a threshold response. So that was an additional 28% increase in urine volume, or another 10 litres per day. The reason we were only feeding up to about 45% of plantain in the diet in this met stall experiment was a bit touched on by Solly's talk earlier, that it's quite difficult to practically obtain plantain percentages more than that in pastures. And then we saw a corresponding decrease in the amount of urinary in concentration. We can see also from this graph that it's not the urine volume is not the only mechanism that's having an effect on reducing urinary in concentration, and I'll touch on this a bit further later. Also in that metabolism store, we actually were monitoring how that urine volume was increased. And what we saw was that individual urination volumes were not increased. It was more an increase in the number of urinations that occurred that day. And that finding was supported by the presentation that Soleil did in her studies at Massey University earlier, which is beneficial because it spreads that urinary N around more of the paddock. Also, the increase in urination volumes tended to happen during the day, and that basically aligns with what we would expect to see. It's correlated with main feeding bouts, and also when basically urination or urine flow isn't being controlled by or not regulated by sleeping behaviours. The main mechanism through which urine volume was increased in the MET-STOR study that we conducted was largely associated with water-induced diuresis, or more plainly speaking, increased intake of water. And what was interesting to note was that increased water intake came largely from increased ingestion of water in the forage material, not through increasing drinking water. And that's largely because, plantain has a higher water content than ryegrass, so the animals were consuming a lot more oh my goodness, I'm going to have to act it out. So the animals were effectively consuming a lot more water in their diet rather than through drinking in the trough. And what was really interesting to note, that when we fed the animals, more than 30% of their diet as plantain, is that they virtually drank nothing from the troughs. And this was an important finding for farm adoption, because in New Zealand, farmers commonly put vet meds or medications through the trough drinking system to mitigate animal health concerns like facial eczema or bloat. So it's important for us to understand that if farmers are integrating plantain into their pastures on farm, they might not be consuming medications that are delivered through a trough-based system. Okay.
Do you remember what your next slide is?
Luckily, I have my notes.
It's all technical hitch.
Oh my goodness. I'm going to go blind.
Yep.
So the final mechanism that has been talked about today earlier as well is a change in nitrogen partitioning.
There you go.
Hooray!
Thank you.
Thank you so much. I'm going to sweat.
I'm going to let you have a little bit longer too.
I don't need it. So nitrogen partitioning is essentially a function of how much the dietary N an animal intakes is converted into product, or how susceptible that dietary nitrogen is to degradation in the rumen. So in the metabolism store study, we fed the increasing levels of plantain in the diet, and as Ina mentioned before as well, this study was isonitrogenous, so each treatment basically had similar nitrogen intakes. And what we found was that as the percentage of plantain increased in the diet, we saw an increase in amount of dietary nitrogen that was partitioned to milk, and we saw a subsequent increase in milk production. And we've seen other studies that have showed elevated levels of milk production or elevated live weight gains when animals have been fed diets with plantain. We saw an increase in the amount of nitrogen that was partitioned to fecal material, and we saw a reduction in the amount that was partitioned to urine. And that at 45% of the diet, it equated to about a 25% less amount of nitrogen that was partitioned to urine, or about 66 grams per day. The mechanisms behind this is that because basically plantain herbage contains favourable traits that alter partitioning away from urine, and some of these include nitrogen fractions, or how the nitrogen in plantain is made up. Plantain contains much less soluble nitrogen in the herbage, and soluble nitrogen is really vulnerable to rapid degradation to ammonia in the rumen. And as we know that, ammonia in the rumen can reach a threshold and it will diffuse out of the rumen into the bloodstream and be processed by the kidneys and excreted. Also, plantain contains a lot more undegradable nitrogen, and in systems where increased undegradable nitrogen would not be beneficial, in systems where dietary N is limiting, that would be a problem. But for our system where we have excessive nitrogen in our forage, it's not an issue. Also, plantain contains higher levels of carbohydrate and a greater carbohydrate to nitrogen ratio. And the work from Ellis in 2001 showed as the ratio of carbohydrates to nitrogen increases, you see a reduction in the amount of urine N that is excreted. And if we superimpose plantain onto this graph, it would be sitting out to the far right. So it's basically that there's more energy available from the plantain forage to the microbes to be utilising that dietary N for either product or integration. Secondly, there is some evidence to suggest that plantain contains secondary metabolites or bioactives that can reduce the formation of ammonia in the rumen. That's some work that Soleil has done earlier to, again, I guess, suppress that ammonia accumulation in the rumen. And Ina brought this picture up too before, but it was just really to show that the diurnal response of feeding plantain is that we do actually, these grey bars here symbolise when we actually presented new feed to the animal in the Metstall trial. And urinary nitrogen, the solid line is cows consuming ryegrass-only diets, and the dotted and dashed lines are increasing levels of plantain. We see that plantain in the diets, we have a consistent reduced urinary nitrogen concentration and consistently lower plasma urea concentration, so that nitrogen circulating in the body is being better regulated. So from all of the work that we've done with the animal studies and metabolism stool studies, we've been able to use this data to adapt the Molly cow model to for New Zealand-based systems and to include plantain in the model. We've used the forage-based input as parameters to develop these models, and have essentially enhanced the model so that we can accurately predict the urinary N excretion from cows fed diets of plantain that might vary in their forage trait composition as either a response to a cultivar effect, climatic growing conditions, or how we manage those pastures. It's a bit of an example of the model. It's probably very hard to see. We've got our cows, Fiona, Deirdre, up there, and this is an example of the model output where the blue dots here are the observed values from 2 Metstall studies, and the yellow and orange dots are what the model is predicting in the various iterations. So it is predicting reasonably well. There's a few outliers there which we'll develop further into, but it's really a cost-effective way of doing some preliminary screening about how changes to the plantain forage might influence nitrogen excretion. And to conclude, relative to ryegrass, plantain has favourable herbage characteristics that reduced urinary N concentration and the inloading of urine patches, which provides an environmental benefit. And the work we've been trying to do is to identify those traits and the levels they are needed in the forage to drive the animal responses, so that we can use this to screen new cultivars for environmental benefit. We can use it to inform plant breeding, like the session that Gemma talked about before, and inform farmers on how they can best manage their plantain for improved environmental benefit. Thank you very much.
Thank you, Elena. We do have time for some questions, thanks to our expert technical assistants down here. Thank you. Otherwise, if it was reliant on me, we'd still be sitting in the dark. So, do we have a question for Elena? Yes.
What percentage of plantain do you need in the diet before you see the
We saw a linear increase in milk production. I guess it depends on statistical difference, what is statistical difference, but when we've explored it in the met stall studies, we did actually see a linear response to improvements in milk production, but we've really been looking at that sort of, to get a substantial response, you're looking at about 30% in the diet. It does depend on what you're feeding it with. Not all studies have shown an improved, a significantly improved milk production relative to a ryegrass-based diet, and I guess it always depends on what you're feeding it with or comparing it to. But we've never seen a negative effect on milk production or growth in livestock-fed plantain.
Have you ever compared it to a ryegrass white clover?
Oh yes, I'm terribly sorry. All the animal studies were ryegrass white clover. Yep.
Over there. Do cows have a preference for plantain?
Yeah, we do get asked this question a lot, and it's very variable because there is certain instances where farmers will say they've seen avoidance of plantain. I've actually had studies, and I should have brought a picture, where I've had the animals actually fossicking through the ryegrass to seek out the plantain. I've certainly noticed in some of our farmlet experiments that certainly, at least up until Christmas, animals will actively select the plantain in the pastures and graze it preferentially. Where I do my research, it's summer dry, and we might see sometime later in the season when the plantain has older leaves, we will see the animals might go for the newer, younger leaves rather than the older per se, but they're probably, the agricom team might be able to comment more on that. We've tried to look at what might be driving some of these avoidance characteristics, and of course when you try and do a study it doesn't happen, but so yeah, it could be related to insect pressure as well, driving up some of these other compounds, but not sure. But it's generally, when it's well managed, it is very palatable.
Well, I'll give Glenn a chance to answer that question while we switch presenters. So, Mike, if you want to come up and set your one up. Glenn, do you want to make a comment, and then we might have time for one more question? Elena answered it perfectly. Okay, we don't need the commercial input then.
Yep. I guess there are examples where animals will avoid this. I think one of them is where we've got heavy seeding, we've got old plants, so that these long rotations where we build up high covers can be problematic. And I think Gemma mentioned that we fear undergrazing more than we do overgrazing. Keeping this in a good round normally eliminates most of those issues, but I agree with Elaine, there are some times that we don't necessarily understand that animals will avoid grazing. There is still some grazing, but you can tell the preferences.
If you can join me in thanking Elena again. Thank you. Right. The final speaker in this plantain session is Mike Dodd, who's a senior scientist in the Farm Systems group at AgResearch in Palmerston North. Mike obtained his PhD in rangeland ecology from Colorado State University. He's been involved in a wide range of research areas over a 33-year career, from pasture agronomy and germplasm development through to systems modelling. He's also currently on the executive of the New Zealand Grassland Association, which is obviously aligned with the International Grassland Association. So Mike, we're in your hands.
And fully supportive, yeah. Well done, Elena, for dealing with that issue. I hope I don't have to, but we'll see. This problem of nitrogen emissions to the environment, whether gaseous or leaching, is clearly a collective problem. While we can demonstrate some really good results within our experimental studies, and as you've seen with Soleil at a farmlet system level, the only way we're going to deal with this effectively is if we get large numbers of farmers adopting this. Technologies that we have available in order to reduce the emissions of nitrogen to environment collectively at regional scales. And so adoption becomes really important. And it's interesting just listening to some of the questions that are very similar to the questions that our farmers are asking us when we present these types of programs, the questions that they have, which are turning over in their minds as to whether they're going to make the leap into this forage plantain. Just a quick nod to my co-authors from Dairy NZ and from Agricom helping to put this presentation together. I'm going to try and make it fairly brief because I think we want to have a good. There's clearly been some good questions, and we want to have a good talk about this. So, firstly, I'm going to kind of go national. And you've heard a little bit of this already from Ina about where we started with forage plantain in the late 90s. And two cultivars which it's kind of a tale of two cities in a sense. One of which was some earlier research was quite unfavourable in terms of its usefulness in grazing systems and had quite a lasting impact on the adoption of plantain. Lancelot, the cultivar, and the other tonic, which has really gone kind of from strength to strength as this kind of story has unfolded over a couple of decades. And I think it's pointed out earlier that much of the research that we have done has been with tonic. But clearly, over the last decade or so, other seed breeders have seen an opportunity in the market as the research has emerged and as the benefits of plantain have emerged, both from an agronomic point of view in various farm systems and from an environmental management point of view. So we kind of saw early on relatively low volumes of seed production in New Zealand. If we go back to the late 90s, 20 tonnes per year. If you compare that with, say, what we were producing in terms of ryegrass, might have been around 13,000, 14,000 tonnes, white clover 3,000, 4,000 tonnes. So really, really small beginnings. And then kind of through that first decade, tested in various farm systems, farmers kind of pushing the limits about what it could achieve. Those volumes increased to quite high levels. And then when those kind of agronomic boundaries of it and factors such as insect pest pressure, where you had monocultures of plantain, a little bit of a retrenchment there kind of in the early part of the last decade. And then coming on a little bit stronger again in the last few years. In terms of the uses that we have put it to, early days, just kind of really a minor component within quite diverse grass and clover-based pastures, and particularly some really good work out of Massey University through the 2000 decade, using it in combination with the likes of chicory and clover in high-quality pastures for finishing young livestock. Then emerging into the dairy sector, only probably recently, really in the last decade, both as a mixed sward and as a pure crop. And I guess if we kind of think about the adoption curve that many of us are familiar with, we kind of feel like we're very much in the early adopter phase at a kind of a national level. So we've had some innovators, they've tested it, some struggles, some successes, and now we're starting to see it emerge into that. And if you look at the kind of a little sort of cheat graph of research publications, you really do see in the last decade an increase in the effort that's gone into researching it. Helped very much by both the current program that we're in and two other programs, I think that Ina mentioned, multi-million-dollar, multi-year programs. So, how at the farm scale, what have we used plantain with? Yes, as a pure forage crop at relatively high sowing rates, but relatively short term, and often as a part of a pasture renewal program going back into mixed grass pastures. And those herb mixtures that I mentioned, dominantly in the dry stock sector with building up ewe tupping weights and lamb finishing, combining with white and red clover and chicory at some more modest sowing rates, but with really probably the best persistence that we've seen in any pastures. And I'm going to kind of say that the lack of grasses in those systems is pretty crucial there, because when you put a grass, particularly a ryegrass and a plantain together, they are going head to head. And then I guess the final one, which I'm going to talk a little bit more about as the talk progresses, grass-based pasture mixes, where it's really a sort of a sub or a minor component at relatively low sowing rates. And the points about persistence that have kind of come up earlier in the session we start to run into those. So Elena mentioned this magic number of 30%. It's not a clear threshold, although the number has been kind of kicked around the sector for a few years now. My observation is that wherever we've done a study where plantain has been more than 30% of the diet, we get good, consistent, statistically solid benefits. Where it's less than that, sometimes you can see an effect, sometimes you can't. But we think that realistically at a farm scale, probably we are going to be unable to get to that 30% level if it indeed, in some of the work that Elena demonstrated, says there is a threshold there, it's not entirely linear. If we're going to get to that level, we're probably not going to be able to rely on our kind of conventional grass-dominant pastures alone. The competitive issues and the persistence issues are too great. So we're going to have to combine different pasture types within a farm system to achieve those sorts of levels. So we did a little bit of a simple spreadsheet modelling exercise to look at varying a couple of different factors to see what it took to get to 30% over the long term, so it's kind of a stable situation for a farm system. Using 3 factors, so the percentage of farm in a pure plantain crop, again probably as part of a renewal program, the regrassing rate, and just for a little bit of context, in the dairy sector, typically regrassing rates New Zealand around sort of 8 to 10%. And then this other factor, which we derived from the Tararua project, which I'll get onto in a little bit minute, looking at what we'll call the decay rate of plantain content in those pastures over time, under a kind of typical average management scenario, and then looking at some of the farmers within our dairy systems that have managed to maintain persistence a whole lot better, which is in the lower here. And you can see relatively quickly that it is quite a challenge to get to, if indeed this is a magic number, and I'm certainly not suggesting that it is, to 30%. And critically, this makes quite a big difference in achieving that. So, what are the sort of enablers and barriers that we've kind of run into? I mean, it's great that we have now a long history of working with plantain from the early days in the late 90s. A lot of agronomic experience out there in the sector. And probably more understanding out there than really kind of sits in the literature. And we've got a group of a kind of farmer population that's really comfortable with establishing what we call homegrown forages. So, reasonably high regrassing rates. So you would think that maybe adding just another component into those forage mixtures wouldn't be too challenging. However, when we talk to farmers about, what would inhibit them, a number of things come up. Weed management, recognising that we are putting a dicot herb into a system where the majority of the weeds that we want to get rid of are also dicot herbs. And so many of the active ingredients that we would normally use to keep our pastures weed-free are a problem. There are relatively few chemicals that are currently registered for use on plantain and mixed pastures. And of course, we are a small country, we have a small market, so any new development of any new active ingredients is probably quite challenging for that small market. And so we really have to work with the chemistry that we've got and fine-tune those systems to achieve that. Pest management, I mentioned before, so some historic outbreaks, particularly plantain moth in the upper North Island, a little bit of a setback for people in using it. We've mentioned already persistence and palatability, so we've had a good chat about that. And then there's this kind of question of regulatory recognition. So on the one hand, you have the kind of agronomic benefits, if you like, the carrot. And on the other hand, we have the stick, which is whether farmers are going to be obliged to use tools like Plantain and other mitigations in order to reduce their nitrogen losses to environment. And there's a certain feeling out there that until Plantain is well recognised as a mitigation by our regulating agencies, then that will be potentially a barrier to adoption. So I'm going to talk about a specific case, a little project that's gone on for the last 3 to 4 years in the Tararua region. So this is on the lower east coast of the North Island of New Zealand. There are about 30 to 35,000 hectares of dairy farms in this region. And when I talk about regulatory management, we have a regional plan that is managed by local government, a regional authority called the Horizons 1 plan, which dictates that farmers will need to have resource consents to carry on agricultural activities in the long term. And one of the key components of that is reducing nitrogen losses to waterways. And one of the key tools that is used in predicting what any given farm system will be emitting is the overseer model. We have a situation where we've got some 180 plus farmers in 2018 at the beginning of this project that did not have a consent to continue farming. So that's pretty challenging, and hence the Tararua rollout project that was initiated. It had some fairly simple objectives so that farmers would be able to achieve consents to farm by adopting best mitigation practices, including Plantain. So this is one of a number of tools that may be available to them. Also that we wanted to appreciate that they would have the confidence to use Plantain and use it successfully. And that, in fact, this was a solution not only for the region but for beyond, and so that those benefits will be communicated beyond the district. So where we went to with implementing Plantain on farm was not kind of a traditional research project, putting a whole bunch of research trials out that were nicely replicated, controlled and balanced, etc., etc. We're dealing with quite a diverse farmer population, even within the dairy sector in terms of their attitudes towards adoption. And so what we did was really push these farmers to use Plantain in the ways in which they wanted to use them. So the types of sward types that I looked at earlier and the types of crop rotations that we talked about. And so we laid on top of that a monitoring program. Yes, there was a little bit of pushing and poking to get some farmers to help us out with certain pasture types that they might not otherwise have chosen. So we put a monitoring program in place over about 3 years to look at the performance of those pastures. Sure. At the start of the project, they had a number of questions. And the first ones were really simple ones around impact on forage supply and impact on forage quality. And so this is the result of 3 years worth of monitoring of various pasture types, mostly in paired paddock comparisons within a farm, but across about 6 different farms, farmers that were willing to put their hands up. And I mean, the essential kind of conclusion that we drew out of this was that if we look at, here's our kind of standard conventional ryegrass clover pasture in terms of seasonal yield, and this is using the Dairy NZ FVI seasons. Just adding Plantain into the mix, the biggest gains seem to be in the summer and autumn period. So here we are, autumn yield, and that translates too into Plantain clover mixtures and into pure plantain crops. So we're getting, in that autumn period, which was mentioned before about the high risk of end leaching, we're getting high plantain contents and increased pasture production. We've already heard about the loss of dry matter as you increase plantain content, kind of along this line. And it was really only at the higher end of the plantain content scale where we were seeing small reductions in digestibility. So we were pretty comfortable to say that no effect on production or quality, really, if anything, a slight gain at certain times of the year. Questions around establishment of plantain into the farm system. So this one looks at various methods of establishment, from direct drilling or cultivation, establishing new pastures with plantain in them, versus establishing plantain into existing ryegrass clover, either by undersowing or broadcasting. And in this location, bearing in mind various climatic factors and soil factors, it was pretty clear that autumn establishment in the context of new pastures was the best way to achieve high plantain contents. It was also the best way to achieve plantain content, the highest plantain content over the long term, and by long term I mean 3 to 4 years. So there was really no interaction between method of establishment and the persistence of those. Sure. Where are we next? Yep. And the final question is one that relates to that regulatory issue, is how can we make sure that we can assess the content of plantain in our farm systems at the farm system level in order to put that into the regulatory documentation that we'd have to supply. And we did a bit of work that really said that you didn't have to work too hard, that visual assessments of plantain content within pastures were actually a pretty powerful tool and correlated well with herbage dissections. So that was a good thing. In terms of outcomes, over the 3 years, we've now got 80 dairy farmers using plantain in the region, which is about 30% of the dairy farmers in the district, and about 3,200 hectares of pastures now with some plantain component in that. Going back to my original number, we're talking about something like 10%. So again, yes, very much in that early adopter stage. And then really the remaining work that sits within the current project that Ian had described to you, incorporating all these effects, functional effects of particularly the soil effects into the models like Overseer, which are crucial for regulation. Practical solutions to weed management options, so getting beyond the kind of off-label use that we're seeing at the moment and getting some solid answers there. And then thinking about, okay, what are some alternatives to ryegrass? If ryegrass is so competitive with plantain, are there other species which we can combine to get a good result? And just finally, some acknowledgements to the various folk that have contributed, particularly to the Tararua project. Thank you, Derek.
Thank you. Well, we have finished on time at 3:30, which means that we are going to head to our afternoon tea break. But anybody who wants to stay and ask some questions, we can facilitate that for a few minutes. But if you want to go and have afternoon tea and refreshments and the poster session, you can go now. But otherwise, we'll answer some questions. Oh, I will actually, before people do start to break, if you can join me in thanking all the speakers, because I think all the presentations are great. So I don't see a lot of people going to the door, so we'll have a few questions. But please feel exit if you want. Questions for Mike from the farm systems approach? Over behind Glenn. What are the economics of putting more plantain in the farming system?
Yes. But if you're going to ask me for some exact numbers, that's going to be challenging. So what's the cost of seed, guys?
It's about the same or a little bit less for a kilo of plantain.
Yep,
A little bit more.
So if we're just adding plantain into our existing regrassing regime, marginal cost pretty low. And if same
price as clover.
Yeah. Yep. So and if these guys are going, say, starting with a pure crop for maybe 18 months and then going into a regrassing, which they would do anyway, marginal cost not huge. Yep. And then if you look at compare that with the value of a little bit extra forage grown in autumn, I think it stacks up pretty well.
So what do you see as the economic benefits from doing that in terms of milk and meat? And
Yeah. Can I turn that around and say that the ability to continue to farm is quite an important economic benefit? And so if this is one extra thing that enables these farmers to get over the line in terms of reaching their in-loss targets, the value is almost infinite.
So it comes
Sorry,
back to the social
that's a little bit of a
licence,
facetious
doesn't it?
answer to the question, but yeah.
It's part of the social licence to operate.
Exactly.
If you haven't got that, and this helps you to get there, what's the value?
Yep.
Yeah, it's a very high quality finishing feed.
So we've certainly seen some plantain dominant pastures, even in the Tararua region, which is a relatively wet region. Really high plantain content, and when the ryegrass is really falling over, that's what you have available. So to maintain milking right up to where you would normally be drying off.
Yeah, I do want to add a little bit to that. With the regional plans getting into place, farmers need to show how they're going to reduce nitrate leaching. So they have to use programs like Overseer or other similar models to show where they're going to go. So we really needed to make a change of mind as well ourselves with modeling economic benefits, because you're really used to compare with what's happening now, but we really needed to change and say what's happening in 10 years. So we're now comparing the cost of implementing plantain with the cost of reducing fertilizer use, reducing stocking rates, reducing production per hectare, and then options like these are highly beneficial.
Thanks, Ina.
Gavin?
I was just thinking on an international basis, other than New Zealand and, say, Ireland, who cares about this?
Pete, do the Dutch, because I know your colleague who was sitting next to you before was telling me that in Holland they are seeing about a 50% reduction. So anybody else want to comment on the international scene? I suppose it comes down to where water quality is becoming an issue, or where you've got nitrous oxide greenhouse gas emissions, Gavin, where you're going to have to be able to demonstrate that on an international scale to produce and export your product.